All right, guys, welcome back. Second video here is chapter 48, all about neurons, synapses, and signaling. And I'm hoping all this is going to make sense. I'm pretty loaded up on pain medication right now, so if I say something that doesn't make sense, that's probably why. All right, let's see here. So, talking about neurons, synapses, and signaling in the nervous system, neurons are the cells that are going to send communication basically from any sensory organ that you have to your brain, and then also from your brain back out to um, your sympathetic and parasympathetic nervous systems, those that are voluntary muscles and involuntary muscles that give your body a response to some type of sensory information that was brought into the body. Okay, so I'm going to jump way down here to this first section talking about neuron organization. Nervous systems process information in three stages: sensory input, integration, and then motor output. So that's the first thing we're going to kind of look at here right away. So sensors are going to detect external stimuli and internal conditions and transmit information along sensory neurons. So the highest concentration of sensory neurons are going to be in your fingertips, in your nose, on your tongue, on your toes, places like that. That sensory information is going to be sent to the brain or ganglia, which are cells within your brain, where interneurons are going to integrate that information. Basically, those interneurons are going to send that information to the right part of the brain to be processed and figure out what's going on with that sensory input. Motor output are going to be neurons that leave the brain or ganglia and then trigger muscle or gland activity. So a response. Many animals have a complex nervous system, usually consisting of two main parts to their nervous system: a CNS, central nervous system, which is basically your brain and spinal cord, and then the peripheral nervous system, PNS, which is just all the other nerve cells that are located in your body, whether they're big or small. So the neurons of the peripheral nervous system, when bundled together, are going to then be called nerves. So if you've ever seen in a book or in a live surgery on TV or something, if they show a nerve cell, it's not a single cell; it's a bunch of neuron cells that are bundled together, and that makes up a nerve. Okay, so a very simple picture of that process. Some type of sensory organ, like your eye, will pick up sensory input. It's going to send it through those sensory neurons to your brain. It will be integrated, figure out what's going on. It will send information then out of your brain, down your spinal cord, to motor neurons that come out of your spinal cord, and then again, finally hook up with a muscle or a gland. Muscles are going to be mostly voluntary. Glands are going to cause an involuntary response. Okay, so neuron structure and function. So neurons look a little bit different than most cells in your body. They have kind of a funky shape. Most of neurons' organelles are going to be within the cell body. Most neurons are going to have dendrites, which are highly branched extensions that receive signals from other neurons. I like to think of dendrites as fingers because you can receive information with your fingers. That's what I kind of think of them as. The axon is typically a much longer extension that transmits signals to other cells at synapses. The cone-shaped base of the axon is called the axon hillock. Okay, so if we have two neurons here. If we look at this top one, here's the body of the cell. Here are the organelles. Here's the nucleus. These extensions are the fingers or the dendrites that come out. They are going to receive a stimulus. You can follow these red arrows. The axon hillock attaches the body to the axon, which is where the information is going to travel, down the axon, and then eventually through what's called synaptic terminals, that information can get transferred to another neuron cell and continue the message. So when we talk about having information sent from a sensory part of your body to the brain and then out to a muscle. It's not one continuous cell that's moving that information, right? There's lots and lots of nerve cells that are passing that information off one to another. All right, now the synaptic terminal, which is the end of the axon, passes information across something called a synapse, which is a gap between the two cells. They're not physically touching each other. And it's going to send that information across that gap in the form of chemical messengers called neurotransmitters. We've talked about these before. So that synapse is a junction between the axon and another cell. So here's a picture of that right here. Here's that gap between the synaptic terminal and the body of that other neuron cell. And it's chemical messengers, neurotransmitters that are being sent across that gap. That's how the message gets moved. And I know I'm going fast. I just don't want my videos to be too terribly long. Okay. So if we looked at the two cells, we have a presynaptic cell and a postsynaptic cell. Presynaptic is the one that has the message first, transfers it to the postsynaptic cell. All right, 3 different categories of neurons. Sensory neurons tend to look like this. They're usually longer and they have a cell body kind of in the middle of the axon. Interneurons, if we're going to go in order here, interneurons are highly branched within the brain because they make more and more connections with other interneurons and parts of your brain. You can see the body right there where the nucleus would be. And then the motor neuron, which we saw in the other picture, looks like this. The body or the nucleus, nuclear area being in the body down here by the dendrites. Okay, here's where it gets tricky. Membrane potential. So ion pumps and ion channels establish what's called a resting potential of a neuron. So every cell has a voltage, which is basically a difference in electrical charge across its plasma membrane. That's called a membrane potential. Hold up, need a drink of water. So the resting potential is the membrane potential of a neuron that is not sending signals. So if a neuron is not sending a signal, it's in resting potential. That does not happen very often with most of your neurons. Changes in membrane potential act as signals transmitting and processing information. So for example, in mammals, a neuron at resting potential, the concentration of sodium or potassium, is highest inside the cell, while the concentration of sodium is highest outside the cell. The sodium-potassium pump, which we've discussed in previous chapters, uses the energy of ATP, because it's an active transport process, to maintain these potassium and sodium gradients across the plasma membrane. Those concentration gradients are going to represent chemical potential energy. So the opening then of those ion channels in the plasma membrane converts chemical potential to electrical potential. A neuron at resting potential contains many open potassium channels and fewer open sodium channels. Potassium then diffuses out of the cell. The resulting buildup of negative charge, because if you're taking positives away from an area, it's going to become more negatively charged, within that neuron is the major source of membrane potential. Okay, so we can look at this from a numbers standpoint. We're not going to. We're going to look at the picture because that's way easier. Okay, so here's our neuron. Here's our dendrites that are collecting information from a different neuron. Or possibly this is a, well, no, this wouldn't be a sensory neuron. This is a motor neuron. So outside of the cell, okay, we have a very high concentration of potassium. Inside the cell, we have a very high concentration of sodium. Wait, no, I said that wrong. Sorry. Outside the cell, we have a high concentration of sodium. Inside the cell, we have a high concentration of potassium. So as those sodium and potassium channels open, okay, we have sodium or potassium that's going to want to move across the membrane to the outside of the cell. And we have many more potassium channels than we do sodium channels. So potassium is going to move faster, which means the inside of our cell, you can see the minus signs here, becomes more negatively charged, and the outside becomes more positively charged. So that is going to correlate with moving a message through that neuron cell. All right, this is just another example using sodium and chlorine, which we're not going to go over. Skip that. Okay, all right, so then action potential is where we actually get the message to move through the cell. Okay, and it all has to do with those gated ion channels that we just discussed. So when gated potassium channels open, potassium is going to diffuse out of the cell, making the inside of the cell more negative. We just saw that. This is called hyperpolarization. Okay, so when sodium diffuses out of the cell, makes the inside of the cell more negative, that's called hyperpolarization, which is an increase in the magnitude of the membrane potential. Okay, so what does that look like? If we look at this on a graph, we can look at membrane potential. Maybe I'll go to this one. Membrane potential measured in millivolts, so it's electric. Okay, and so here's our resting potential of our cell. Here's where we're at, time in seconds. Hyperpolarization makes it more negative, so we drop. Okay, that's a hyperpolarization. Opening other types of ion channels triggers what's called a depolarization, a reduction in the magnitude of the membrane potential. So for example, depolarization occurs if gated sodium channels open and sodium diffuses into the cell. So what does that look like? Okay, well, it's the opposite. We have more of a positive increase in that potential, so it's called a depolarization, which seems backwards, but it's depolarization, means you go up on the graph. All right, so graded potentials are changes in polarization where the magnitude of the change varies with the strength of the stimulus. So these are not the nerve signals that travel along axons, but they do have an effect on general nerve signals. So let's look at if depolarization shifts the membrane potential sufficiently, it results in a massive change in the membrane voltage called an action potential. Action potentials have a constant magnitude and are a all or none phenomena and transmit signals over long distances. Okay, so you can see we have this threshold line, which is for all neuron cells. If the membrane potential in millivolts reaches that threshold or surpasses it, you will have a huge jump in your membrane potential, which is called an action potential. Okay, so that's going to move that message through that cell, no questions asked. Okay, so if we jump and I'm going to jump ahead here to the final picture. If we wanted to walk through this graph and show it what's happening in the cell, start down here. Here's the resting state of that cell. So this is in resting potential. The inside of the cell is more negatively charged than the outside of the cell. That's what you should remember. Depolarization, okay, is where those sodium ions are going to start to move into the cell. Okay, sodium ions are moving into the cell, and we are closing the potassium channels so that they cannot move out. So that's going to cause depolarization, a more positive increase in the cell, depolarization. K3 then is going to be the rising phase of the action potential, where more of those sodium channels open up and those potassium channels are still closed, causing a more positive increase in the inside of the cell, getting us past the threshold. Number 3, and getting our action potential. Well, once we've reached our action potential and the message has gone past that part of the membrane, we then go to a falling phase of the action potential where we reverse what channels are open. Potassium channels open, sodium channels close, and our membrane potential reverses where the inside of the cell now becomes more negatively charged than the outside, and we have a huge hyperpolarization. Now, as that falls, you get to where we have something called an undershoot, where as the channels start to close up, there's going to be more molecules that fit through and cause a problem with the polarity, or I should say the potential of that membrane, and so it shoots a little bit lower than resting potential, and then some of those channels open up to kind of balance it back out to natural resting potential when there is no message going through that part of the cell. So this whole picture, is happening at multiple places along that cell membrane, down the axon, and through that neuron cell. So this is definitely a graph that you are going to want to know. There is something called a refractory period. So after an action potential, a second action potential cannot be initiated. In other words, that part of the membrane needs to basically reset itself to resting potential before a new action potential can come through, before a new message can come. The refractory period is a result of temporary inactivation of those sodium channels. I'm laying here in bed doing this and my arm just fell asleep. All right, conduction of action potentials. Let's jump down to the pictures here. Okay. So like I said, along the membrane of this axon, you're going to have the message that starts to move. Sodium and potassium channels are going to start to open. Right now they're opening here. This is where we have a lot of the action going on. Nothing is really happening here. Okay, this is in resting potential, still in red. So if we move that message along, the blue is going to indicate our action potential. This is where the message is located. In front of that is the resting potential still, and behind it is going to be that refractory period where it is on the graph hyperpolarizing. Okay, there's going to be some undershoot, and eventually it's going to get back to resting potential. So again, this is a good picture to know as well. Okay, adaptation of axon structure. So what makes axons so great as part of a cell? The speed of an action potential, so the speed that a message can travel through those cells, increases with the axon's diameter. So it's kind of like the bigger, it's like a fire hose compared to a regular hose that you have at your house, a small hose. The more water that can go through, the faster you can get that water through and get the message across. In vertebrates, axons are insulated on the outside by something called a myelin sheath, which causes an action potential's speed to increase. So myelin sheaths are made by glia cells. They're also called oligodendrocytes in the central nervous system, and Schwann cells in the peripheral nervous system. So here's what that looks like. It looks like a roll of toilet paper. There are these flat, thin cells that wrap around the axon that help move that message. So the more myelin that you have, the better your nervous system works. As people age, they tend to lose myelin, and so their movements of messages do not travel as fast or as far, sometimes not at all. You have these spots called nodes of Ranvier. Action potentials are formed only at nodes of Ranvier, which are gaps in the myelin sheath where voltage-gated sodium channels are found. So if I go back to the picture, here are these nodes of Ranvier. They are the gaps between the two toilet paper rolls. And so as the message moves, okay, you can think of it almost like hopping from one node of Ranvier to the next, because that's where the message is going to depolarize and start over. Okay, lastly, let's talk about how neurons communicate with other cells at synapses. So we talked about how from cell to cell, there's going to be a gap between the synaptic terminal of one cell and the body or dendrite of the next cell that has to get the message. And that message is passed through neurotransmitters. So as the message comes down, these vesicles are going to form around these neurotransmitters. They're going to be released through exocytosis and travel across that gap and hopefully be picked up by ligand-gated ion channels or receptor proteins in the membrane of that other cell. And this is what it looks like up close here. So that's going to cause a chain reaction, a communication reaction. We also have calcium that can travel into that other cell, helping it to move those neurotransmitters. Okay, so we can have postsynaptic potentials that fall into two categories. Excitatory postsynaptic potentials, or EPSPs, are depolarizations that bring the membrane potential toward threshold, or inhibitory postsynaptic potentials, IPSPs, are hyperpolarizations that move the membrane farther from the threshold. Okay, so we can look at examples of that. So after release, the neurotransmitter can kind of really do three things. They can diffuse out of the synaptic cleft and be picked up by the next cell. They can be taken up by surrounding cells, or they may actually be degraded by enzymes. So not all neurotransmitters actually get picked up by the appropriate cell. All right, let's look at some examples here. So, for example, if two EPSPs, excitatory postsynaptic potentials, are produced in rapid succession, there's an effect called temporal summation that occurs. Okay, so two rapid successions of message that are going to move, you can get temporal summation. So in other words, you get a boost of membrane potential and then another boost, which gets you past the threshold and gives you that action potential. That's called temporal summation. So you can see the green here. You have two quick bursts of message, basically, that are coming into that cell, which causes an action potential to happen. Okay, another example is called spatial summation, produced nearly simultaneously by different synapses on the same postsynaptic neuron added together. So this can trigger an action potential as well. So again, here's a postsynaptic or a terminal here, and here's a separate one going to the same receptor cell. If they both go at the same time, you can get enough of a membrane potential increase to get you to the action potential. Okay, here, if you have one come in and one comes in through what's called an inhibitory postsynaptic potential, it can cancel each other out. So meaning that you're not going to hit that threshold. Because you have to be able to stop messages from being sent as well. You don't always want the same messages being sent all the time in your body. So you have to have excitatory and inhibitory working together. Okay, I think that was it. Here, one more thing. Some examples of neurotransmitters you should probably be aware of. Acetylcholine, amino acids, biogenic amines, neuropeptides, and gases. If you could just recognize what some of those are, you'll be just fine. You don't have to know the molecular structure or anything like that. So these last few slides here give you just some examples of what those neurotransmitters are used for in different organisms. All right, that is it for chapter 48.